Stable. So welcome everyone to the November Bristol BioDesign Institute webinar. In terms of like administrative things, so if anyone turned up a little bit late, I'm going to post into the chat again the link to a website called Slido, which we use for questions. If you go onto that website and then type in the number 79179, you'll be able to actually post your questions in there, or you can upvote other questions that other people have asked, and then myself and Claire Grierson will field those to Domitilla at the end of the talk. If you include your name, then we can mention who exactly it is that's asking the question, if that helps with any kind of clarification and things. So to start, I just want to say it's a real pleasure to have Domitilla Del Vecchio here today. Domitilla is a professor in the Department of Mechanical Engineering at the Massachusetts Institute of Technology, and also a member of the Synthetic Biology Center they have there at MIT. Domitilla's background is actually in dynamical systems and control theory, and she obtained her PhD from Caltech, working with Richard Murray, and then moved to the University of Michigan, Ann Arbor in 2016 as an assistant professor, and then later on in 2010 she took up her position at MIT in the Department of Mechanical Engineering. She's had numerous awards, and a few of them include the NSF Career Award in 2007, the Donald P. Ekman Award from the American Automatic Control Council in 2010, and more recently was a Bose Research Award participant. In terms of her research, it's really great to have her because Domitilla is renowned for basically developing or attempting to develop methods to enable a more kind of modular design of biological systems. And in doing so, she's revealed a number of really interesting aspects of biology, such as things like retroactivity, or the really crucial role that resource allocation plays in biological systems. But what I think probably defines Domitilla's work is the kind of rigorous theoretical underpinnings that it has, and the way that she uses often quite simple experiments to really demonstrate many of these effects actually occur in real systems. But what's really nice is that she doesn't just demonstrate these effects occur, she also uses that theoretical understanding to build new devices that can overcome some of these challenges. And so it's a really nice demonstration of the power of bringing theory and biology together, I think, in terms of actually making biology more rationally engineerable in different ways. And so it's a real pleasure to have her here today. And today she's going to be speaking about context dependence of biological circuits, predictive models, and engineering solutions. And without any further ado, I'll hand over to Domitilla.
Thank you very much, Tom, for the great introduction, and thank you for inviting me for the seminar. It's a very exciting opportunity. So today I will present some of the work we have been doing in the past several years on issues of context dependence and actually modularization of genetic circuits. So I will start out with a brief overview. Probably most people are familiar with this concept, but as an introduction, I just want to point out that the opportunities that engineering biology offers are tremendous. And the field is really based on, the technology of genetic engineering. And in around 2000, that's when the field was coined, right, as synthetic biology. There was about a decade where people were focusing on building simple motifs to understand biological mechanisms. But there was an exciting transition from my point of view around 2010-ish, where people started realizing that, well, we can connect more complex systems together to create functionalities that are so sophisticated that perhaps one day we may be able to mimic what this neutrophil is doing in your bloodstream. Right? When I saw this moving, I was really excited because this is a great example of a sensing, a computation, and an actuation in a feedback loop, which works extremely reliably. And so the notion is that with engineering biology, what if we can re-engineer all the sensing, computation, and actuation components to create novel systems that solve problems, right? And here I'm just showing a few examples of what this applications can be. For example, tracking, recognizing, and killing cancer cells, right? This is ongoing research in a few labs where people have been programming bacteria so to localize at a tumor location, and once they're there, they can sense, detect it, and decide to lyse open and release killing agents. Applications that are much newer are those regarding the control of cell fate, so controlling the way mammalian cells make decisions about specializing in one type or another. And then there are other applications that are more based on bacterial and cell-free synthetic biology that really deal with detecting and counteracting threats, for example, biological sentinels that detect pollutants or threats that can be detected, for example, very efficiently by bacterial cells. Virus detection, fortunately, is very popular nowadays, and there are fantastic opportunities in that field as well. But that said, I will go right to the point. So when we build these biological circuits, right, independent of the application, we typically build them bottom up. We start from a library of parts, promoters, ribosome binding sites, coding sequence, and then we assemble that into functional modules that can be, right, oscillators, such as the repressilator, or toggle switches, such as Jim Collins' toggle switch, or more sophisticated logic gates. And then we assemble together to create more sophisticated functionality, for example, chemical signature classifiers, right? And then we put them in the cells and hope the cell to be able to run the circuit, right, to produce the right functionalities. And this way we can program a cell with new functions. Modularity is critical in this process because it allows to manage complexity and time. And for this, it's absolutely necessary that the behavioral component is independent of surrounding systems, right? In practice, however, modularity fails. So a component's property are often affected, as many of us know, by surrounding systems. So the problem this way is that anytime I'm building a big system and I'm adding a new component, all the components that are there need to be redesigned from scratch. And this leads to a very painful combinatorial and error-prone design process. Very lengthy, and for a circuit, for example, of 10 or 11 genes, it takes a PhD thesis, right, to complete. So in this talk, I will focus on a couple of sources of context dependence. This is in no way exhaustive list, but some sources that we have been focusing for a long time is one is the issue of loads. So anytime you connect an upstream system to a downstream system, it's no surprise that the downstream system is applying a load to the upstream one and therefore is changing its dynamics. Something that more recently we started looking at is the fact that all the circuits that we are building, they are running in the cell. And they're using the cellular resources. So we can't forget of the fact that cellular resources are finite. And if you're loading the cellular resources with your circuits, you may create actually quite interesting interactions that were completely unexpected. And then, of course, there are many other context issues. Cell growth is a major one, and off-target effects, right, are other key players. And so all of these are sources, I would say, of uncertainty that affect your genetic circuit when you build it, and they affect actually the ability for you to predict accurately how the behavior is going to go. In this talk, I will focus mostly on the cellular resources problem. And in particular, I will give you some examples just to give you a sense of how this can play a role, and then I'll dive into the essence of the talk. So this is one first example that came up in our lab when we were designing bacterial cells for biosensing. In biosensing, you often want that your cell is able to sense multiple molecular species coming from outside, and this is a very trivial multi-input, multi-output sensor where you have two completely independent sensory paths that go from U1 to green protein, from U2 to red protein. This is just an example of a genetic implementation. So given the independence of these two sensory paths, what you would expect is that when you have only the U1, you will have green. When you have only U2, you have red. And when you have both together, you expect to have both green and red. If you do the experiment, however, that's what you find out. So you find out that there seem to be some crosstalk where one sensory path is affecting the other. In this talk, I will try to convince you that many of these problems can be explained by accounting for the fact that these circuits are sharing resources that are limited. And I will specifically address transcriptional and translational resource sharing. Another example that are still coming from our lab, and again, we bounced into this by chance when we were building a simple AND gate. Okay, so an AND gate is a system that takes two signaling molecules as input and gives an output when both of them are present. A very simple way to design this is to have a cascade of two activators, each one inducible, so that when you have both inducers, both red and green are high. So this is what you should expect, that you have an increasing function when both of them are high. And so the predicted behavior of this is that when you only have one of the signaling molecule, you have a low output. When you have both of them, you should have a high output. So we did an experiment. This was back in 2015, was the first AND gate that we were building for a system we were considering. This is what we got. So it's clearly not an AND gate. It's actually doing the opposite of an AND gate. And at this point, given the simplicity of this system, I was very disappointed with the student that was working on this, because I said, we are talking about curing cancer with genetic circuits and we can't build an AND gate. What's going on? So the students were very frustrated, clearly. So he went back to the drawing board and he came out with a hypothesis that if I look at this circuit in the context of the cell, I have that RNAP and ribosomes are required for the gene expression process. So when I apply an input, a chow in this case, okay, I am essentially diverting all these resources in the first stage of the cascade, so less is left in the second stage of the cascade. And therefore, what he showed me is that you can actually have an increasing, but then a decreasing portion of the overall dose response curve, because in this part, the resources being sequestered by the output outcompete the strength of the direct activation. So I thought this was very interesting. So we're starting to investigate this further. And this goes the essence of the talk. So I will spend the first half of the talk on predictive models that we have been considering for this phenomenon. And then I will dive into engineering solutions to somewhat isolate circuit behavior from context. So we went back to the central dogma, essentially, of molecular biology and to the way activators and repressor in bacterial cells affect expression. And of course, the standard model, which is a textbook model, shows that the rate of production of any protein is proportional to this regulatory function, which is often called Hill function. And then this function can be increasing if the regulators are activators, or decreasing if the regulators are repressors. Something interesting that is often assumed is that there are some constants in front of this Hill function, which are actually proportional to the amount of RNAP and ribosomes. but these are not constant variables. So these follow a conservation law. So the total amount of RNAP and ribosomes is conserved. So if you take this into account and put it back in this model, you will find that the true regulatory function is not just your hill function. Something more complex pops up. In particular, there is this term that pops up that shows that any protein PI is going to be affected not just by its intended regulators, but also by any other regulator that you have in your system. Specifically, for example, if UJ is an activator for PGA, the hill function is going to be increasing, and as a consequence, since it's in the denominator, it's going to repress the PI. The strength of this hidden repression is directly proportional to what we called the resource demand coefficient, which is proportional to the DNA copy number, okay, of your regulator DNA, to the RBS strengths and to the promoter strengths of the target that it's regulating. So this is telling us that new interactions emerge. So if we call this G the new regulatory function, we have that the effective interaction graph is now composed of one portion, which is the same interaction that you had before, but they are rescaled, so they are becoming a bit weaker than they were before. And then there are hidden interactions that appear because of this essentially denominator popping up. So in summary, here's what we are finding out. Anytime you have a transcriptional activator, it becomes an effective repressor to any other protein, because in the act of activating its target is sequestering, right, resources from any other target. Similar, if PJ is a repressor, it becomes an effective activator for any other node, because in the act of repressing its target is freeing up resources for other proteins. And then we have rules that show us that the intended interactions stay unchanged in sign, although become weaker. And then there are some cases where the outcome depends on the parameters. So these first three cases here, the rule is completely independent of the biochemical parameters. That's what it is. But in some cases, those are parameter dependent and needs to be studied case by case. Given that, essentially, we did a bunch of experiments, right, to confirm this, what we call lateral repression. So we had a gene, the red, that we could activate. We had another gene, the green, that was constant. And what we showed that as you increase the expression of the red gene, or for what it matters of any other gene, right, the green constitutive goes down. And this was showing about 70% effect in a system with medium DNA copy number. Something very interesting that we saw is that once we were decreasing the strength of the ribosome binding site of the target to almost zero, we didn't see almost any effect on the PI, so the GFP. This is telling us that, at least in bacterial, in exponential growth, the main responsible for these interactions are translational resources, which is going to be really important when we go to design. You can work out the math, and you can actually show that there is a rule that tells us that any two proteins expressed in bacteria need to lie on a line. So this is what theory predicts. If I'm expressing any two proteins, they're always going to be constrained on a line, and the slope is dictated by the price of the proteins that depend on the ribosome binding site strengths of the corresponding genes. If you take the data and you plot it on P1, P2, you see quite a nice line, and you show that you can tune the slope by changing the prices via modulating the ribosome binding sites. So that said, we went back to our broken activation cascade. So if we take into account the effective interaction graph, meaning the hidden interactions, we see that there is this repression that comes up due to this ribosome sequestration. And its strength, it's proportional to the DNA copy number and the ribosome binding site strength of the target of the protein, so of P1. So what we did then, we created a parameter space where we could modulate J, the resource demand coefficient, via DNA copy number tuning and ribosome binding strength. So J small means essentially no resource competition. So we are working in this regime of the parameter space. So this interaction, the red one, is really weak. In the top right, it's the opposite extreme. The resource demand coefficient is really high. Resource competition should be dominating the behavior of the system. So then we created a library of cascades where we could tune the ribosome binding site strength and the copy number so that we could move in this parameter space. So the first experiment that we did was here, and that showed a biphasic behavior of the input-output cascade. So that was an intermediate strength of this hidden interaction. Then we increased the DNA copy number to demonstrate that we actually break completely the cascade. Okay, so the cascade now becomes almost like a repression cascade. Then we started to try to fix it. So we decreased the ribosome binding site strength, and we went back to the almost desired nominal behavior. We then decreased also the DNA copy number strength to obtain a very small J, and we obtained back the monotonic increasing response of the cascade, which is what we expected. So this is to say that if you're building a system, you can use J as a guidance to minimize the strength of this hidden interaction to recover, okay, the intended behavior of the system. So I just want to add one slide to let you know that mammalian cells are not immune to competition for resources, but there is one major difference with what happens in bacteria, and that's that the resources being competed for are mostly transcriptional resources. So here is a simple experiment which is identical in concept to the one we did in bacteria, where we are activating a red gene here, and we are watching a blue gene, which should be constitutive. So as we are activating our target, we see increased activation, but at the same time, we see this lateral repression due to resource competition on this theoretically constitutive gene. Something that we were also curious about is whether these effects would be particular to a specific promoter or cell line. So what we did, we created a library of this circuits with different promoters in different commonly used cell lines. And we saw that essentially the behavior of resource competition is essentially present in all cell lines across all promoters. So purple in this plot shows that the nominal output is getting decreased when essentially your target is activated. So that said, I just want to spend the second part of my talk in going through engineering solutions. And in particular, here is the two design approaches that I'm going to present today. So first of all, my view is going to be the control systems view of how we can make systems robust to perturbation. So I will view my genetic device as a system which takes inputs, regulators, and gives output proteins. And then resource changes, for example, fluctuation in the level of ribosomes, or untargeted effects, or unexpected binding, any of that, I will view that as a disturbance to my genetic device. That can happen at the transcriptional, the translational level, we get into there. So the expectation is that this device, when the input is presented, the output is going to suffer and change with an error compared to the desired one. So there are essentially two main ways in which you can address this. One is negative feedback control. So the idea is that instead of applying a constant input to your genetic device, you make that input change based on the error that the output is experiencing due to the disturbance. So that's going to be your feedback controller. And the idea is that the compensation action of the feedback controller should cancel, okay, the disturbance influence. A different type of approach, which I'll also cover, is feed-forward control, which is also used in engineering in most scenarios. Feed-forward control is essentially finding a way to sense, okay, the disturbance and do some pre-compensation so that you are canceling effectively the effect of the disturbance on the output. Whether you're using approach 1 or approach 2, if you design them well, you should expect that your desired output should go back to its level, okay? So in normal engineering systems today, in most cases, you have a combination typically of feedback and feed-forward control. So I'll show first the feedback control approach. So what we are going to do in this case, we are going to consider D, our perturbation in the free level of ribosomes, and design a quasi-integral controller that ideally should allow us to reach back exactly the desired level after some time, right? So it's called adaptation. Okay, how do you reach adaptation? Well, the basic approach is to use integral control. So you should be able to apply a corrective action, okay, to your process, which is proportional to the integral of the error between the true output and a desired reference output. So the main challenge in implementing integral control, which is something that we have been working on for a while, is leakiness. So bacterial cells are growing quite fast. And so there's always a decay that you can't avoid in any protein or molecule dynamics. So what we then thought is that we can't send a growth rate to 0. So what we are going to do, we are trying to make the compensation action of the controller much faster. So essentially, if epsilon is really small, so meaning we can make this error correction really fast, then it's almost as if we can neglect this dilution term, okay? So the outcome of this is that for a molecular implementation, we should consider all fast controller reactions that compute the difference and integrate, okay? I just want to point out that for this to work, the theoretical underpinnings are not too trivial, so you can check the papers for that, but I won't go into those details in the talk. So the implementation that we chose for our integral control was via sRNA silencing, and it's shown here. So we have our protein, and its level is fluctuating when ribosome availability drops, so which I'm depicting here as a disturbance D. So what we do, we have this protein produce an sRNA that targets the mRNA for degradation. If you write down the ODEs of the system, you can find out that the controller reactions, which are involving M and S, are naturally very fast. First of all, RNA interactions from M and S are really fast, so we can get naturally a 1/epsilon there. And then you have high RNA transcription rates, which we can engineer in both promoters by picking strong promoters. Bottom line, you can demonstrate that you have the memory variable here being M minus S, and therefore you have this quasi-integral structure with fast dynamics that should work. So here's the implementation in bacteria. So on the right-hand side, I'm showing a resource competitor. This is the system that allows us to apply the disturbance D in ribosomes. So we are overexpressing this red protein, and in doing so, we are taking away ribosomes from the production of GFP. Then, as you can see here, we are producing GFP co-transcriptionally with an ECF, which is then activating our sRNA and targeting the whole mRNA of GFP for degradation. So due to the post-transcriptional nature of this controller, I just want to point out that the inputs and the outputs of your device are unchanged, so they are untouched by the controller. So here is the experimental result. On the left-hand side, I'm showing an unregulated device where the sRNA is not there. So it's an open loop system. When we apply the perturbation, so AHL is being applied, we produce RFP, then we have that the output goes down. In the right-hand side, instead, when the controller is present, the GFP goes down, but then recovers through this common adaptation response. Distributions are controlled as well. So as you can see here, and also as you keep increasing the perturbation AHL, you can see that the open loop system suffers more, but the closed loop system is quasi able, right, to completely reject the perturbation. So I just want to show that it's absolutely critical to have fast reactions in your controller. If we slow down the reactions by, for example, slowing down transcription rate of our RNA species, you can see here that the robustness score drops by almost 100% to around 60%. Okay, so this is just demonstrating that the principle of fast reactions is absolutely critical in these types of controller to overcome dilution. So the second type of approach that I'm going to show you is feed-forward control. This is the case again where we have our genetic device. D is also going to be the change in the availability of resources. And then we have to be able to design a sensor that pre-compensates the effect on the output. So the way the design works is that we have an endoRNase, which is a protein that can recognize a target sequence on a target mRNA and partially cleave that. Okay, so essentially it's an enzymatic reaction where the eRNA binds with the mRNA and targets that for degradation. So you can see here that if I look at the resources being either transcriptional or translational, they come in both paths, so that what you should expect here is that once you are activating competing modules that are applying this loading on ribosomes, resources go down, but then the ERN expression goes down, so the output is de-repressed. And this de-repression should compensate for the drop in resources through this path. So we engineered the system in order to be able to tune the key parameters that control performance. So the key parameter that control performance in the system is essentially epsilon. So epsilon is telling us how large is the undesirable operation regime. Ideally, if this is the resource level and this is the output should be completely flat. So we would like to have a small epsilon, so to increase the desirable operation regime. Epsilon can be decreased by increasing the Ca-C, okay, which is our picked endoRNAs for the design. We know that epsilon is already naturally quite small because the catalytic rate of Ca-C is known to be quite high. We therefore built a system where we could tune the level of Ca-C by upstream open reading frames. And what we saw is the following. So on the left-hand side, we show the fold change in the output for an unregulated system, where instead of Ca-C, we have FLUC, which is an inert protein. And on the right-hand side, we show the regulated system, where we have Ca-C with changing numbers of UORF. So moving this way, the level of Ca-C is increasing, and you can see that for the highest Ca-C level, we have essentially adaptation. You can also compare an open loop and a closed loop with comparable output levels. You can see that for the same comparable output level, you can create a feed-forward controller that stabilizes the output for all input perturbations, while the unregulated output still goes down. So that brings me to the final comment that I want to make, that we tried to operate this feed-forward controller in a bunch of different cell lines and across a bunch of different activation domains for our regulator and promoters, okay? And as you can see in the bottom here is that the purple areas are much less than in the top. So the top here is the open loop system, and the bottom here is the closed loop system. Okay, so this is to say that this device is easily transportable across cell lines, and it seems to work reliably and stabilize expression across cell lines. So that brings me to the end of my talk. As a short summary, I have gone through the issue of resource competition. I've shown you how in bacteria, translational resources, specifically ribosomes, are a critical resource competed for. And I've shown you a post-transcriptional controller to mitigate that effect. In the case of mammalian cells, transcriptional resources that are actually mattering the most for the circuits. And I've shown you an incoherent feed-forward loop that can stabilize output expression independent of resource fluctuations. There are a bunch of other things going on in my group, which I won't have the time to talk about today, but I'm happy to chat with people offline if they're interested in hearing more about them. So I just want to thank the students that did the work. All the bacterial work was done by Hili Hyun, who got his PhD and now is doing a postdoc. He's a fantastic student. He designed all the controllers and did also some of the experiments. Sino Wang is our lab manager that supervised all the project. And Ross Johns is also graduated and doing a postdoc, and he's the monster of the mammalian work. And I'll finish there. Thank you.
Thank you very much, Domitilla. That was a really, really nice talk. I can see in the Slido, so if anyone has any questions now as well, please keep adding them to the Slido, and we'll try and get through as many of them as we can in the time we've got left. So I don't know, Claire, is there any that you wanted to pick out to start with?
There are lots of good ones here, so but I think I'll just start at the top. So, Domitilla, do you think natural biological systems exploit these types of effects, or resource sharing effects? So have they arisen during evolution? And so I think let's just start there. There's another part to this question, but I think let's just stop there and think, do you come across any examples in the literature? Or?
Yeah, so I think that those issues of essentially coupling systems indirectly through some upstream shared resources is a motif that is found in many natural systems. And actually there is some rather beautiful work done at Princeton by the Schwarzman group, who have traded this motif specifically in the MAPK cascade. Okay, so I've been talking about gene expression here, but we have a lot of work done also in signaling pathways. And this is a very common motif there. And in the Schwarzman lab, they have investigated that in vivo in the natural system. So definitely, there are many of these cases happening in natural systems. A second example that I know people have been considering quite a bit is this issue sometimes referred to as docking sites or decoy sites. So it turns out that many transcription factors have binding sites on the chromosome, some of which are with high affinity, but they don't have regulatory effects. And so there are quite a lot of people that have been investigating in natural systems, why would that be the case? And we now know that having all those downstream sites that are competing for this upstream transcription factor that is also binding target is affecting the dynamics of the transcription factor and its response. So that could be an additional way in which you can tune the response of a system without changing its parts.
Yes, I think this is really interesting because it might help explain some things which have often seemed like redundancy, for example,
Absolutely.
in biological systems, where it's actually not redundancy, it's about modulating levels and competition for resources.
Absolutely.
Yeah, very interesting. The second part of that question was about whether there are specific examples where anyone's exploited any of these natural systems. I don't know if you know of any.
So
in engineering particularly, I think.
So I am aware, I'm not sure where the publications are, but I am aware of a stream of work that was looking at how to tune the, for example, the period of a synthetic genetic clock by adding binding sites for the transcription factors. In fact, there are very interesting consequences of on the period of the clock if you're adding binding sites for that. And I can search some of the references if people are interested. So that's one first example. The other example, I think that there was some early literature around 2011 where people were trying to exploit the fact of adding a lot of off targets to a transcription factor to make an on-target response more or less ultra-sensitive. So I think that was also with the notion of engineering a system using this effect.
Okay, we've got a lot of questions, so I will move on quickly. Somebody asked, both of these are anonymous, how the biomolecular controllers impinge on the resources. And I guess the question there is how you build that into your design. So when you're adding molecular controllers in and they might be impinging on resource models, you need to put those into your, I mean, I think that's kind of taken into account in the modeling you're already doing. But is there any element where that needs refining?
So is that a question of when I design the controllers?
I'm reframing the question really. It asks about the resource costs of the molecular controllers themselves and how they impinge on resources. So it's about a design question about whether you take that into account and how you take it into account in the designs.
In the design of biomolecular controllers, the resource?
Yeah, the resource costs, yeah.
Yeah, so, oh, I see probably what that question is. So if I may rephrase the question, my understanding of that is that different biomolecular controllers may be using themselves resources for their, yes, that's absolutely true. So for the sRNA based, since the key component there used is an mRNA species, it's not using ribosomes. So from that point of view, that's somehow a little bit lighter on the resource loading that we are trying to defeat. On the feed-forward controller, the feed-forward controller on the opposite is exploiting the fact that the feed-forward branch, it's using exactly the same resources, so it can pre-compensate with the opposite effect on the output. So there are somewhat different requirements, whether you're doing a feedback or a feed-forward.
Okay. There's a question about RNA polymerase competition, saying that people have tended not to think that it's an important issue in E. coli, but your theory seems to suggest that a limitation on RNAP is the cause of the isocost steepening as the ribosome binding sites are weakened. Would you agree with that?
So, okay, so our data suggests that for the extent of competition, that we have seen, the ribosome competition is the major player. So I would say that we quantified it out of 100% of the effect, probably 10, 20% or even less is due to transcriptional, and the rest is due to translational.
Okay, that's really helpful. Was a question about how you modified translation initiation rates in mammalian cells rather than E. coli.
Yeah, so it's using URFs. So these are upstream open reading frames. So those have the ability to, so essentially more, and I can provide the reference that to that. It's a paper of 2 years ago. They show that if you add open upstream reading frames, you can almost linearly decrease the translation rate of your output protein.
Great. Staying on RNA-related topics, with increasing complexity in synthetic systems, will it become more necessary to include more RNAi regulatory elements to act as rheostats and reduce noise?
Yeah, so I think a question, right, when you go up to more of this genetic modules in the cell, I mean, my impression is that using, on the one hand, parts in your controllers that are lighter in the sense that they use mRNA processing as opposed to protein processes may be more beneficial, maybe for scalability. There are some downsides, which I haven't mentioned quite in the talk, in using mRNA regulation as opposed to protein regulation directly, which is the fact that if you have any perturbation, not ribosomes for sure, but any perturbation acting directly on the protein itself, for example, you have off-target effects on that protein, with this mRNA-based feedback, you wouldn't be able to compensate for it.
Okay.
So I've got a question that I've just sort of picked out in here, and something that I'm quite interested in is one of the things I thought was quite fascinating when you were showing the differences between the bacterial like resource that seems to be maximally limiting, and then in mammalian cells, they're kind of completely different processes in the cell, and whether there's some kind of inkling as to why that might be biologically. Like, is there a complete difference in the way the resources are used that might accommodate that or account for it?
So yes, actually, this is a fantastic question, and we spent a lot of time thinking about it. In bacterial cell, the process of transcription is very simple. Essentially, you have the RNA polymerase that is almost the key resource required. In mammalian case, in addition to the RNAP, you have a bunch of auxiliary factors that are required for transcriptional initiation, for example, the mediator. And then you have a bunch of additional transcriptional cofactors that are required for the elongation process. So the transcriptional process there itself is using many more resources than bacterial systems are using. And what we have found by digging in the literature is that people have been investigated specifically through also in vitro experiment that the mediator seems to be a key transcriptional resource that it's highly competed among genes and transcriptional activators. In our experiments, where we also looked at the mRNA levels, we found that probably only about 20% or less of the competition we will see will come from translational resources in mammalian systems. So it's kind of the opposite of bacteria. And we are still debating, it's an open question, I don't have an answer, of whether the fact that, for example, the mRNA is shuttled outside the nucleus is providing some form of compartmentalization and isolation. So the short answer is that we don't know.
No, that's fair enough. Because I was thinking whether there might be it might be one of those kind of evolutionary transitions that you want to move away from one situation to another because it gives you more access to regulating things in different ways, maybe, I don't know. But I do think it's a really interesting outcome of what you found in these results.
Yeah, we were very surprised too, so.
There's a broad biological question in why you're seeing a translation limiting in microbes, but transcription limiting in mammalian cells, and whether that has something to do with the different objectives of those cell types.
I mean, that has most likely, right, an explanation along what the objective of the cell has. I would also say that there are probably many functions that the ribosomes are performing in bacterial cells that perhaps they are not in mammalian cells, and especially the energy and growth regulation in mammalian cells is so much more complicated. There are the mitochondria, right? In ribosomes, in bacteria, they're essentially taking care of the whole thing. So I think there is a lot of to study there and ask questions, and that's definitely something that we're interested in understanding better.
That's an interesting bit of cell biology to think about. Yeah.
Definitely. And I had a question. So the sort of the systems that you presented were kind of like small bolt-ons to a cell, like a slightly new functionality. But obviously they were self-regulated, but they're within the context of the cell, which may not be regulated in the same way. And are there, as these systems get bigger and bigger, are there going to be kind of detrimental effects on, say, the endogenous regulatory mechanisms that you would need to take into consideration? You'd need to understand better to make sure you're not like inadvertently doing something bad that the cell then can't function just generally in some way.
Yes, so that's absolutely a critical question. So in fact, when we were looking at, in the bacterial work, at the negative feedback controller design based on sRNA, we were particularly concerned in looking at the growth rate and if we were going to affect in any way, right, the growth rate with the controller or without the controller. Of course, in our system, we used that controller on one gene only. So it turned out that in that case, we didn't see any growth effects. Of course, you're asking the key question here, I think, if what happens if you're adding that to multiple genes, right? What's going to happen? So we have some theoretical work that just appeared on arXiv that is trying to understand what's going to happen if you have multiple of this doing this selfish feedback regulation, right? And somewhat fighting with each other. And theory predicts that you can come up with quite brutal failures, unfortunately, if you do not design the system well. So that's all we know so far.
Well, that's very interesting.
Yeah.
So a couple of questions have been upvoted while we've been talking, so we could definitely ask them as they're getting support. The first one from Davida Salzano: what are the advantages of feed forward with respect as opposed to feedback approaches? I mean, I imagine feed forward is faster because it's anticipatory. Is that roughly right?
That's absolutely true. So for sure, apart from the fact that feed forward, it's easier to build and tune. I mean, it was probably 6 months work to get that feed forward to get built and function and tune. For the feedback, it took much longer. And absolutely the case that in the feedback, there could be some slower dynamics in the feedback
more
loop
oscillations, I guess.
That may lead to some form of oscillations, and it depends a lot on the delay you have in the feedback path. So you have to be very careful to that. So I would say that's probably the major difficulty, right? So there are more, I would say, constraints that you need to make sure are satisfied when you do the feedback design, as opposed to the feed-forward design. However, for the feed-forward design, if there is a small change in the feed-forward path, the system is going to be going off. So essentially, it's not going to be robust to perturbation in the feed-forward path, okay? While in the feedback configuration, if you have some perturbations in some of the parameters of the controller, you probably can manage better robustness there.
That's a very thorough answer. Thank you, Domitilla. The next one is from Gonzalo Vidal. Reducing the transcription translation costs of outputs implies work with less particles, making stochastic effects more relevant. Is there a relevant trade-off between cost and noise? So,
okay, so the first part to the answer is that in all of our devices, open loop or closed loop, the output level was the same. So we never compare an open loop system and a closed loop system where the open loop has very high level and the closed loop has a low output level. We always tune parameters so that they are the same to make sure the comparison is fair. For sure, if you are decreasing the output in a design that goes to very low levels, stochastics may play a role. In our designs, we were always working at quite high molecular counts.
And we're very short of time now. We've got less than a minute left. Should we? Do we want to fit one more in, Tom? Have you seen one?
I'm going to be really selfish. I'm going to ask a question that I've been sort of thinking about as you've been talking about things, and that is, do you think so? The models you showed were quite simple, which is nice because you can do nice analytics on it and really understand what's going on. Are there more complicated aspects of biology that you're overlooking by abstracting it in that particular way, which might be more important for some types of system we might want to build in the future, these really complicated ones that you alluded to at the start?
Yeah, so I think that our approach as control theories, as you're saying, is always start using the simplest model so that you can get a sense of the concept, right? So what's the key concept there? We are definitely finding out in the specific applications that we're working on self-fate reprogramming via genetic feedback controllers, that the designs that we are considering now, they're way more complex than the ones that we initially thought. And that's because, as you're saying, there was a lot of the biology that we weren't really thinking of at the beginning. And in a situation where you're really tampering with the decisions that the cell is making, so you're playing with the endogenous regulatory network of the cell, it's full power, then you're absolutely right that we are seeing that those biological details are not actually details. Yeah.
Yeah, that's cool. I think that's really, it's interesting because obviously you don't want to have to put all the details in initially because you need to understand, like you said, you need to understand the principles of it. But yeah, I think is important.
Yeah, I think simple models are good to give you a good starting point of how to reason about things. So I would say that the final concepts may not be that different from the initial ones with the simpler model, but the implementation, right, may be much more complex because it takes care of additional details. And then sometimes you also find out that the concept needs to be changed, right?
Definitely. So I think looking at the time, we've still got loads of questions, but I'm really sorry, we're going to have to kind of draw a close there. I just really want to thank Domitilla again, and also Claire for helping to field the questions.
Well, thank you, Tom, too, and Domitilla.
Yeah.
Thank you very much.
Very much.
Everyone for coming. And in terms of the kind of format for these seminars, we're looking to kind of restructure them in the new year. So I'll be posting some things online to let people know a little bit more about that. But again, thanks, Domitilla. It was brilliant talk.
Please come and visit soon because there's so much we could
I would love
about.
to. Anytime, really. Thank you. It was a pleasure. Bye.
Bye-bye.
Bye-bye.